Hello, everybody, and welcome to our fascinating medical sciences lecture titled Defeating Pneumonia and Meningitis with Vaccines. Thank you very much for all of you joining us today. My name is Seita Diop. I am a current MSci Medical Innovation and Enterprise student here at UCL and your chair for today's event. Today's fascinating lecture will address how powerful vaccines can be and consider the necessary steps to further protect us from pneumonia and meningitis. We want to keep these lectures as interactive as possible, so please make good use of the Q&A function on Zoom and submit your questions throughout the session for our speaker, Professor Rob Heidemann. The hashtag to use on Twitter for this event is hashtag FMS Lectures. That's hashtag FMS Lectures. I am delighted to now introduce our speaker. We have Rob Heidemann. Professor Heidemann is an infectious diseases doctor and a scientist. His research bridges working with patients, cutting-edge laboratory approaches to working out how and why infections occur, and on disease prevention. This work focuses on life-threatening infections caused by so-called mucosal pathogens, which are bacteria and viruses that live at the back of the nose and mouth. As Institute Director, he transformed the highly successful Malawi-Liverpool Wellcome Programme into a centre of excellence led by Malawian and international scientists, improving the health of people in sub-Saharan Africa. Since moving to UCL in 2015, Prof. Heidemann's work has continued to centre on understanding the mechanisms of disease and optimising the development and implementation of vaccines to prevent severe infections in the UK and in Africa. Thank you so much, Rob, for joining us today. And now I'll hand over to you.
Thank you very much, Seita, for a very kind introduction. Vaccines are really essential tools for the prevention of life-threatening infections. They're particularly important for preventing diseases of poverty. In this lecture, I will talk about the successes, how we're trying to improve the impact of vaccines, and what we still need to understand. We've clearly come a long way from the early days of Edward Jenner, where he took an extract from a cow's udder and vaccinated a child, to the present day. And what I hope to do is to convince you that we're on the road to defeating pneumonia and meningitis with vaccines. So what is pneumonia and what is meningitis? So pneumonia is infection of the lungs in which the tiny air sacs, the so-called alveoli, fill with fluid and pus, so that it's difficult for oxygen to get across from the lungs to the blood, and that clearly makes people ill. Meningitis is infection and inflammation, sometimes with pus, of the protective wrappings of the brain that go around the brain and the spinal cord, and it's usually caused by infections. This causes brain malfunction, brain swelling, and clotting of the brain. The important thing about the bugs that cause meningitis pneumonia is that most of them live at the back of the nose, and in most people don't cause any disease. But particularly in the very young and the very old, and people with immunity, which is not quite as strong as it should be, and in the context of environmental pollution or viral infections, they get from the nose into the lung to cause pneumonia, or get into the blood and then to the brain to cause meningitis, or sometimes directly from the blood to the lung again to cause pneumonia. The important thing about this process is that you need carriage, and that's carriage of this pneumococcus in the context of a whole zoo of bacteria that live at the back of the nose in order to cause disease and then to be transmitted to others. And it's also important to remember that for the bug itself, causing severe disease in terms of pneumonia and meningitis is largely a dead end. So it's not to the advantage of these bugs to cause death in their host. What they really want to do is to be carried and then be transmitted to other susceptible individuals. Pneumonia and meningitis are globally important diseases. Pneumonia is a leading cause of death across all age groups. Around 22.5 million people in 2019 succumbed to pneumonia, and is the leading cause of death in children under 5 years of age, the most vulnerable in our society. This is a picture that I took of a wall in the school in Uganda, showing the lungs and actually the sort of air sacs where pneumonia occurs. And bacterial meningitis, although less common, there are approximately 2.5 million cases of bacterial meningitis across the world each year, is associated with a much higher rate of death. So around 10% of people with meningitis will die overall, and based on data from 2019. And the other thing about bacterial meningitis is that particularly in West Africa, it can be associated with epidemics of disease. And this is an image taken in Malawi of a patient with meningitis who has had the fluid around his spinal cord and brain sampled with a so-called lumbar puncture in order to make a diagnosis of meningitis. And as you can see from these photographs, these are diseases that often occur in the poorest populations. And this is illustrated by this map here, which is taken from what's called a meningitis tracker, which was developed by the Meningitis Research Foundation. And this can be accessed on this URL. And what this map shows is the level of disease across the world, where the darker colors are the highest burden of disease, and the lighter colors are the least burden. So when you compare the UK to sub-Saharan Africa, Central and South America, and parts of South Asia, you can see that that meningitis, and here in this case neonatal sepsis, are occurring in the poorest populations. And within sub-Saharan Africa is a very small landlocked country, Malawi, where I was based for 8.5 years. And I'm going to use quite a lot of evidence generated from Malawi as to illustrate how we have set about trying to defeat pneumonia and meningitis. As I mentioned, Malawi is a landlocked country dominated by a beautiful lake, one of the Rift Valley lakes. I was based in the south of the country in the commercial center, Blantyre. The capital is Lilongwe. The population of Malawi is now around 20 million, although when I went to live in Malawi in 2005, it was more like 12 million. So it has a huge population growth. Life expectancy is now around 65 years of age, which is still low compared to the UK. HIV still is relatively common in Malawi, around 8%, although when I first went to Malawi, it was 18%. And the reason that HIV has been controlled so well in Malawi is because Malawi has had one of the leading public health approaches to HIV control, not only getting drugs to the people who need it, but also in reducing mother-to-child transmission of HIV. Still a prevalent disease, but much lower than it was previously. Childhood mortality rates are still high compared to the UK, 10 times higher, but much lower than they were in previous years. And again, death in childbirth still remains a major problem for mothers in Malawi and other sub-Saharan African countries. As you can see, 40 times more common than in the UK. So a poor population with a high rate or high burden of disease. And this is where vaccines can have their greatest impact. And indeed, Malawi has really been at the forefront of vaccine introduction in order to drive down a disease caused by vaccine preventable disease. Now, the evidence that I'm going to present to you over the next 15 minutes or so has been generated in Malawi by a partnership of international and African scientists, really dominated by real expertise and skills of laboratory scientists, doctors, nurses, and field workers, both in hospitals and in the community, who are based in Malawi. And I think this is an important message to get across, is that this is evidence that has been generated locally. So being based in the large commercial center of Malawi, in a very large hospital that sees tens of thousands of cases of severe illness every year, The centre where I was based was able to monitor meningitis and pneumonia over time. And one of the first pieces of evidence that was generated from the institute where I was based was that a particular cause of pneumonia meningitis, Haemophilus influenza type B, or Hib for short, which is now present in all the vaccines that are given both across sub-Saharan Africa, but also across Europe and the United States, that this a particular cause of meningitis and pneumonia was controlled by the introduction of vaccine in Malawi in 2002. This is time across here against the number of cases of meningitis seen at the hospital where I was based in Malawi. And you can see in the data collected by the Institute in this red line that it went down dramatically in the early 2000s with the introduction of vaccine. So this was some of the first evidence in sub-Saharan Africa that the vaccine could reduce the burden of meningitis and subsequently pneumonia. What you can also see is that one of the other major causes of meningitis and pneumonia, a bug called Streptococcus pneumoniae, also went down. Now, this wasn't caused by the direct effect of the vaccine, but actually by other public health interventions that were going on in Malawi at the time. So the control of HIV, improved nutrition, improvements in child poverty, all went together in this very poor population to have an effect on pneumococcal disease without a vaccine, because this vaccine doesn't work in pneumococcal disease. So the question is can we control this further with pneumococcal vaccine? So the pneumococcus, or Streptococcus pneumoniae, SPN, otherwise known as the pneumococcus, is an organism like Hib with a thick outer wall capsule. And there are more than 100 types of SPN, rather than with Hib, there's just one type. There are more than 100 types of SPN. And this thick outer wall capsule possessed by the pneumococcus forms the basis of the vaccine. We can't create a vaccine for all 100 types of pneumococcus, and so we focus on those most likely to cause disease and most likely to be caused with antibiotic-resistant infection. So currently, there are vaccines against 10 types, PCV10, 13 types, PCV13, and recently developed, which has just been introduced for adults, PCV20, based on this thick outer wall capsule of the meningococcus. Now, when the capsule was originally used as a vaccine, it didn't work well in small children. But cleverly, what it was found is that if it was linked to a particular protein, in this case from the diphtheria vaccine, it then became potent even in very small children. And the real challenge was that although a single vaccine will protect against one capsid type, you needed multiple vaccines. Indeed, you needed 7, 10, 13, 20 of these vaccines in order to protect against meningococcal disease. So not just one vaccine, but 7, 10, 13, 20 vaccines all in one. So it makes it a very expensive vaccine. So very cleverly, these were generated, and then these have been widely introduced across the world, including Europe, the United States, and across the world. This is a map generated by the Johns Hopkins IVAC consortium, showing that the vaccine in green has been introduced really across the world, with notable examples such as China and some countries in Africa, but most of the world, including Malawi, has introduced the conjugate vaccines, the PCV, so-called PCV vaccines, in order to combat meningitis and pneumonia caused by the pneumococcus or strep pneumoniae. So in rich countries, this vaccine has worked incredibly well. So this is data from the United Kingdom collected by what was then the Public Health Laboratory Service, but is now the UK HSA. This is the number of cases of the disease per 100,000 over time. The first vaccine, which only had 7 capsid types, was introduced in the early 2000s, and then subsequently a 13 type vaccine was introduced in 2009, 2010. The red line represents the amount of disease caused by the capsid types in the PCV7 vaccine. As you can see, following introduction of the vaccine, it again, rather like I showed you with Hib, went down very rapidly, but there was still some disease caused by other types of pneumococcus. So then a 13-valent vaccine was introduced, or a vaccine against 13 different types of pneumococcus was introduced. And again, that went down over time following introduction. So a dramatic direct effect for children 2 to 4 years of age. But what was remarkable about this vaccine is that it also had an effect in those who hadn't received the vaccine. So this is again a similar graph, but this time for people over 65 years of age, so for older population. Now, these people had not received the vaccine, but what we're seeing in these older people was that following PCV introduction, vaccine types went down, and then this was further accelerated by the PCV13 vaccine. So this was an indirect effect or herd immune effect, whereby the vaccine, by reducing transmission from very young children to their older parents or grandparents, reduced disease in those populations without the necessity for actually vaccinating that target population. For the eagle-eyed view, you will also see that there's a purple line that's been going up. And that has been the problem in both young children and in older adults, is that although we're protecting against these particular types of pneumococcus, other types have been going up in the meantime. But this is a demonstration of herd immunity. So what is herd immunity? So when I say vaccinated, I mean those people that have received vaccine. When I say immunized, what I mean is those that have been vaccinated with a vaccine, but also those that are protected. So those that are people in this community that are in yellow are those that have been immunized and are protected. Those that are in gray color have not received the vaccine, and those in the red color have got disease and are able to transmit the disease to other people. So in a population with a low rate of vaccine in the community, the bug in people who've got disease can then be spread to other people and cause disease in those other people. But what happens is that as the level of vaccination goes up within the community, those in the herd that have not received the vaccine or have received the vaccine but don't respond well to the vaccine are protected because there is so little transmission, because so many other people are vaccinated. So this indirect protection prevents those that are infected transmitting to those that are most vulnerable in the community. And that's what happened in the 65-year-olds in the United Kingdom. So the question is whether in populations where the disease is so much higher and the prevalence of the bug is so much higher, will these vaccines also be protective? So this is data that we've generated from Malawi. This is following the introduction of the vaccine in the late 2011. This is over 10 million years of observation. So this is a very large population in data that we collected in Malawi, looking at the disease in young children and in older people. The vaccine was introduced here. This is the amount of disease in the population. You can see that the amount of disease has been going down even before the introduction of a vaccine. And the question is has the vaccine had an effect? And to decipher this, what we've used is mathematical approaches that have actually been used for financial forecasting to really understand the effect of vaccine on these populations. And what we showed is in those types that are affected by the vaccine, in children 1 to 4 years of age who've received the vaccine, disease rates were 74% lower amongst those children and 80% lower in 5 to 14-year-olds some of whom have received the vaccine and some of whom may not have received the vaccine. So there is some herd immunity or indirect protection. But disappointingly, there was very little evidence of herd protection in older people older than 15 years of age. So the vaccine had a direct effect on those that received the vaccine, but much less of an indirect or herd immune effect on vulnerable people who were older. Nonetheless, the vaccine did have dramatic effects in the population. So one of the effects that it had was it actually reduced death rates due to all causes in the population. So what we did is looked in very large populations, actually outside Blantyre, elsewhere in Malawi. We're able to demonstrate that deaths not caused by road traffic accidents or by trauma, but caused by other diseases, all reduced in populations that received the vaccine. So clearly, by preventing pneumonia, you were also making the population more healthy, so less susceptible to other diseases. And that's really important when you're trying to make an economic case for a vaccine. So next, we started to wonder, why isn't the vaccine as effective as we've seen, particularly in richer countries? We undertook a series of studies to try to address this. What we found is that in a series of studies looking at the carriage of the bug that causes pneumonia and meningitis in the back of the nose, is that the vaccine hasn't controlled carriage of this bug at the back of the nose as well as we had hoped, and certainly not as well as in the UK or the US or in Europe. The other thing that we found is that the vaccine schedule that was being used worked well in generating antibodies, so the part of the immune system that controls disease, but that immunity waned very quickly, such that by the second year of life, children were less protected. And then in complex genetic experiments of the bugs that cause meningitis, we found or identified pneumococcal families, particularly those that were resistant to antibiotics, that appeared to be escaping vaccine control. So that all of these things together were leading to less good control, particularly in these high disease burden populations in Malawi and indeed elsewhere on the African continent. So the next question comes, can we improve the control of pneumococcal disease? And a number of groups around the world are really focused on trying to improve the control of pneumococcal disease and therefore the control of meningitis and pneumonia. This is including testing alternative scheduling of the vaccine, and we're undertaking that currently. Testing smaller amounts of vaccine so that the vaccine can go further and can be used in the second year of life. And also in studies that are being led by a colleague of mine, Brenda Kumbwana Adams, in Malawi, targeting very young children by actually boosting the immunity of older children to improve herd immunity and protect the very young in society. This is so-called cocooning. So if you boost the older kids, you prevent transmission of the bug that causes disease to the very young before they receive the vaccine, and therefore reduce disease in the most vulnerable. And therefore, and in addition, there are a whole series of groups testing alternative vaccines and vaccine approaches, including groups here at UCL who are actually studying whether you can, in some way, make the bug less likely to cause disease, but use it as a vehicle to improve the immunity against pneumococcal disease. To complement these activities, we've been working with communities across Malawi to develop cartoons and animations to develop discussions around improving vaccine confidence so that more and more people take the vaccines. And indeed, there is a great deal of confidence in the pneumonia vaccines in Malawi, but also to improve an understanding of how when you give the vaccines, they can also improve herd immunity. So give that indirect protection to those most vulnerable in society. Here at UCL, we've also been trying to understand exactly this whole process of carriage of the pneumococcus, or this pneumonia-causing bug at the back of the nose, and its prevention with vaccines in the laboratory, and also, as I will mention in a moment, in humans. So in the laboratory, we take cells and study their interaction with the pneumococcus, which is shown in green to the red cells here, both understanding what happens on the surface of cells as the bug passes between cells, and also as the bug enters in the cells that are in the nose. And we've also used electron microscopy to understand the intimate interaction between the bug and the surface of cells. Here you can see the pneumococcus on the surface of the cells. Here you can see it moving into a cell. You can see a sort of a cellular ruffle here. And here you can see actually a bug below the surface of the cell, almost like a sort of blanket resting over it. So this enables us to study its interactions and think about how better to control this process. And here in this particular cellular model, you can see the hair-like processes on the surface of the cell, and other cells within the nose cells producing mucus, better known as snot to other people. But here, when these cells are infected, you can see the bugs on the surface of the cell, but also you can see big globules of snot being produced. And that snot may be important for clearing the bug from the surface of the nose, but also may be important for transmission, because any of you in the audience who are parents will know that the first thing that a child or a baby does as soon as its nose is snotty is wipe it on their parents or their grandparents, therefore facilitating transmission potentially to other vulnerable people. So we're studying the bug in cell models, but we're also studying the bug in human experimental models, whereby we deliberately infect healthy individuals, both young individuals and older individuals, in collaboration with colleagues in Liverpool and now in Oxford who are leading this work. And this enables us to study the intimate relationship between the bug and its human cell. And here you can see in green, these are bugs on the surface of the nose of somebody in samples that have been taken. The blue dots are nuclei, the sort of a genetic processing part of a cell. Here you can see a blow up of a cell. Here it's nucleus, and here you can see the bugs on the surface of the cell. These bugs are getting into the cell, and these bugs are moving through the cell. And this is enabling us to better understand the process of carriage of the bugs that cause meningitis and pneumonia, and then develop ways of interrupting this process. And then finally, the other activity that we're involved in is working with policymakers, particularly WHO, to develop worldwide approaches to defeating both meningitis and pneumonia in the coming years. And we've been lucky enough to be involved in developing a WHO roadmap for defeating meningitis with a target of 2030, and particularly spearing the bring together of a global meningitis partnership, which is actually looking at the genetic material of the bugs that cause meningitis in order to use that to understand the disease, understand where it's causing it, and develop better vaccines. This is the WHO roadmap for defeating meningitis by 2030. So I'd really like to stop there by acknowledging that this is work of a huge number of people from really across the world. The work that I talked about in Malawi has really been led by colleagues that include Todd Swarthort and Tandy Mwalekomo, Rosaline Farouk and Comfort, who really led a lot of the studies that I described in Malawi, in Bridget Dennis's lab in Malawi. I would like to acknowledge my long-term collaborations with Neil French and Kondwani Jambo, and the community engagement work that's been really led by Roderick Sambakunzi, and also colleagues at the Malawi Ministry of Health that have really facilitated the work that we've done, which is now being led by Aku and by Brenda in Malawi. I should acknowledge the work here at UCL, led by Caroline Waite and Elizabeth Chan, with fabulous input from Andrea and Rory, and our collaborators here at UCL and at Liverpool and Oxford. This work was funded by a whole series of donors: Wellcome, Bill and Melinda Gates Foundation, the UK Aid and NHR, and also by the Medical Research Council. So I'd really like to stop there and thank you for your attention.
Thank you so much, Rob, for that. That was a really fascinating lecture, and I personally know I learned a lot from what you were talking about. I would like to just say to the audience once more, if you have any questions, feel free to submit them through the Q&A, and Bob will be happy to answer those. But we'll kick off with some of the questions that we've received so far. The first one I have is a question on herd immunity. So it was, oh, sorry, not herd immunity. The question is, why, if they've noticed that in sub-Saharan African countries, they are known to have a higher population density than in some more developed countries, cities, or districts. Do you think this could be a reason why some countries are more at risk of pneumonia and meningitis?
Undoubtedly. I mean, pneumonia and meningitis are diseases of poverty. Even if you look in the most richest countries, like if you took the United States, for example, these diseases are more common in poorer populations, even within the richest countries. And undoubtedly, population density plays a role, because these are passed from person to person, either by coughing or sneezing, or by direct contact with fingers or other things infected from touching your nose. And that happens much more commonly in densely populated places. And certainly these diseases are much more prevalent in the poorest parts of the world, as I showed you, so particularly Sub-Saharan Africa, South Asia, and parts of Central and South America.
That's interesting to know. So it's really an intersection between, poverty, and also incidentally that it causes people to likely live closer together. So the two things are paired. Okay, that makes sense. So the next question we have is, as you were talking through your presentation, we noticed that there was a peak of streptococcal pneumonia in 2011. Do you think there's a reason to that, if of course the reason is known at the moment?
So I think if we're talking about the peak in Malawi, I think that's where the graph started. So there was a huge volume of disease then. What's interesting is that the disease was coming down anyway, and that's true in many societies. As societies control diseases better, you get less crowding, you get some economic improvement, diseases get improve. And many diseases in the UK, for example, went away before the introduction of antibiotics and vaccines. But what vaccines do is accelerate this process greatly. And as I showed you from those figures, we're still anywhere between 10 and 20 times, these diseases are any more than 10 or 20 times more common in many African countries than they are in the UK. So the burden of disease was very high. It has come down with vaccines, but it's still not at the level that we would like. And that's why we're trying to improve vaccines and improve the way that we give vaccines.
I think that leads nicely onto the question that we have here about kind of the challenges in achieving herd immunity. We have a question here saying that it's quite hard to achieve herd immunity across a large geographic area. The question is, is there a concern that maybe the pathogens would mutate at a rate faster than we're actually vaccinating the people to achieve herd immunity? And if that is something that we're concerned about, do you think there are any methods to prevent this or kind of mitigate that risk?
I mean, it's a huge worry. And if you take Haemophilus influenzae type B, the bug that I talked about before, because there's just one type, it really hasn't had the chance to mutate and then come back very much, because we got in there quickly with vaccines. We were able to largely eliminate it from the population, although not completely. The problem with the pneumococcus is there are 100 types. We can only make vaccines to a limited number, and they're very expensive, these vaccines. There's huge investment, particularly for poorer countries, to provide these vaccines. And so there is a real risk of vaccine escape. And so this is why we are trying to develop better ways to really drive these down so that they can't escape vaccines. But you're right, it is a serious problem. Herd immunity develops if you can get beyond a certain level within a community. As I think, it has been demonstrated with the COVID vaccines, it's been impossible to achieve herd immunity. But with diseases that cause pneumonia and meningitis, for many of them, we are able to get vaccines to a level where you reduce transmission and therefore protect the most vulnerable. And if you can do that very rapidly, then you may be able to prevent that vaccine escape. But constantly, we're trying to develop new vaccine approaches to counteract those things. So it's a constant war, and clearly, bugs don't have thought and aren't more clever, but they're certainly more adaptable than we are. So they are changing very quickly. But there have been huge successes nonetheless.
I think the successes are definitely a part to focus on sometimes. It can feel a bit like a game of cat and mouse with the diseases that we're trying to eliminate. So the success are definitely something to champion and celebrate, I guess. So our next question here is, you've talked a lot about the Haemophilus influenza B vaccine. And the question is if you know whether this has been introduced in the UK, or if not, if there's a perspective that it might be in the future.
So it was introduced 10 years before it was introduced in Africa, which I think was a great shame, really. It should have been introduced at the same time. And that's maybe a discussion we can have at another time, why vaccines always get introduced into richer countries first and then to poorer countries later. With the pneumococcal vaccine, the second vaccine I talked about, that they were closer together introduced, but nonetheless, there was a time lag. So Hib was introduced, in the early 90s. So anybody born after about 1992 would have received the Hib vaccine, and that program has been hugely successful in the UK. Hib has been largely eliminated. There are just a handful of cases in the UK every year.
I think that's always that could be a whole other session in and of itself, about kind of vaccine inequalities and everything about that. But no, that's a useful answer. We have a question here from Yulia wanting to know, how can we determine between correlation and causation when we're looking at the effects of the PV13 vaccine? And how do we know that the overall national health is improving as a direct result of these vaccines?
So you've probably been talking to the reviewers of our papers, I would guess, because that is often a discussion that we have. Clearly, we're demonstrating association. We're not necessarily demonstrating causation, although there's a very, I think, very clear link. You can demonstrate those accelerations. And as I mentioned, in these large population studies, you can apply actually tools that have been developed for financial forecasting to demonstrate that there is a likely effect. If you want to demonstrate a mechanistic effect, the human challenge model is quite a nice way of doing that. So when we deliberately infect an individual, you can give them a vaccine beforehand and demonstrate that it actually does protect them. And that certainly we're able to do. And also in the larger scale population studies, when we introduce a vaccine where the population is vaccine naive, you can demonstrate protection under those circumstances. So you're right, in the population studies, these are associations rather than definite causality, whereas in vaccine trials and in the human challenge model, you are able to demonstrate causality. And what's great about the human challenge model is it's a great tool in demonstrating proof of principle in quite small populations, so that then you can then test things in much larger populations.
This is a question from me, so Neff, who's moderating, don't be angry, this is from me. But I'm actually curious whether you think that kind of how do I word this? come back to my question. Let me ask someone else's question. I'll come back to that one. We have another question that's asking, is it possible to create a vaccine that works against the shared characteristics of every streptococcus stereotype, sorry?
So there are various approaches that are being taken. As I mentioned, the current vaccines are all use the capsule of the pneumococcus as its vaccine antigen linked to a protein to make it more effective. And the problem is you keep generating more and more and more and more, but actually, as was asked right at the beginning, the bug is clever and is able to evade immunity. And you're talking, I mean, the current pneumococcal vaccines are around $64, $65 a dose. And that's hugely subsidized in order to deliver them to the poorest countries. But that is ultimately unaffordable if you keep adding more and more vaccines. And the other problem is as you increase the number, because, you know, essentially the current vaccine we're using in the UK is 13 vaccines in one, and you know, the 20 is 20 vaccines in one. Ultimately, what happens is as you add more and more vaccines, there's a danger that some of the other components don't work quite so well. So people are trying to develop vaccines against proteins in the bug. And as I mentioned, colleagues here at UCL, led by Jerry Brown, is trying to develop a vaccine approach which involves a weakened pneumococcus that's alive but doesn't cause disease to induce immune responses. But there are difficulties in the pipeline to that thought and that research getting through to generating a vaccine that then is taken up by a commercial company and then is put into the arms of the people that most need it, because it's very difficult to do the trials, because it's not like you could compare your new vaccine with nothing, because you've already got a very effective vaccine. And so comparing your new vaccine with the existing vaccines is also quite problematic. So the route to licensing those sorts of approaches is quite problematic and something that we're trying to work through at the moment. But you're right, we do need alternative approaches, and it may be that we'll need a combination of vaccines rather than one type or another.
And my question just came back to me. So thank you for that, first of all. The question was about if maybe kind of accessibility to information is part of the conversation that needs to happen. I think lectures like this are amazing and people can, from all over the place, can tune in and find out a bit more. But I think at least some of the papers that I've read, if you're not familiar with some of the terms that you're using, it's all very jargony, lots of numbers, lots of stats. How do you think kind of your everyday person can engage with the data that's out there that supports the use of vaccines without feeling overwhelmed or kind of inequipped to understand what the data is that's out there?
So I'm not going anywhere, but I'm just reaching into a box. I'll be 1 second.
Is this a Blue Peter moment? Here's one I made earlier.
Yeah, so indeed. So we've been fortunate enough to be funded both by the UK NHR and by Wellcome in quite a large public engagement program to develop a dialogue between communities and researchers and policymakers. It's called our All for One program. It started in Malawi. We're also developing dialogues in South Africa and other African countries, and also starting to make the connections to also connect schools in the UK with schools in African countries to generate a conversation around vaccines, how they work, how they might be improved, to improve confidence in vaccines. So this is the comic that we've generated, One for All for One. I'm sorry, it's backwards. And we've also developed animations. I showed you Roderick, who's leading this project. And this is really developing that conversation around vaccines. And it's really been humbling, the insights that communities, even from the poorest African populations, have had into how vaccines work and how vaccines protect communities. And that's really enabled us to build confidence around the vaccines that we use. And as I mentioned, Malawi has been one of the very early adopters of many vaccines. And I think it's been a real pity how social media has sort of eroded people's confidence in vaccines, even in a very poor country like Malawi, But initiatives like this, and there are many like these around the world, are really trying to sort of generate that dialogue and improve that conversation so that people don't feel distant from the facts and what's really happening.
I think on that point, I think I should say, first of all, like, congratulations on that. I think it's such a big step forward in getting people engaged in those conversations and making them feel like they're taking value from the conversations that are happening and moving forward, more empowered to take on their health and like engage with the system of healthcare. So yeah, that's really amazing. So another question from our audience: are nasal sprays or salty nasal washes helpful in reducing the number of pathogens? And do you have any nose washing tips?
I wish they made a difference. I don't think there's really any strong evidence that they reduce your risk of pneumonia or meningitis. But what you will be interested in is that there is increasing interest in delivering vaccines through the nose. And that's because, we're getting to the point where we're putting so many vaccines into injections. It's hard to sort of think, what else we can add. And as I've mentioned, for these pneumonia meningitis bugs, they actually live in the nose. And so if we can develop better immunity in the nose as well as in the blood, that is a possible way forward. They proved very difficult to generate. There is already a flu vaccine, for example, that's given to children. And so that's one potential route for administering protection without having to have needles, particularly in poor populations. But I think we're a little bit away from that to do with the pneumococcus. We are engaged in some work to use particular bugs that don't cause disease, but are very similar to other bugs that cause meningitis, to try to induce protection. So deliberately infecting people with good bacteria in order to protect them from the bad bacteria.
It's interesting that you touch upon that. I think I've heard about certain diseases that are like incidentally protective of others. So that's quite an interesting topic that you touched upon there. Okay, so our next question: how do you proffer the risks versus the benefits of the vaccine? So again, touching upon that kind of education around vaccines question again.
So I think it's important to recognize that all vaccines have side effects, but I think for all of the vaccines related to meningitis pneumonia, the benefit far outweighs the risk. The risks are minimal. These vaccines have been put into the arms of children and adults across the world, many millions, with minimal side effects. So I think the benefits far outweigh the risks. And I showed a picture of Edward Jenner vaccinating a child, and the risks of that smallpox vaccine were considerable in those days. And in fact, the original vaccine that was used with us was smallpox itself, which indeed did harm some people. But I think we've come a long way from that in terms of our ability to assess the safety of vaccines and assess their efficacy. So the vaccines that I've been talking about have huge sort of safety, good safety profiles, and the benefits far outweigh the risks of these particular vaccines.
I don't know if you'd agree, but I think it'd be fair to say that that's probably true of most healthcare in general. To some extent, there is a side effect, but we administer what we do because the benefits far outweigh, like you said, the side effects that are involved with healthcare and medicines in general.
Yes, I mean, I fully appreciate that vaccines have a special case, because what we're doing with vaccines is giving them to somebody that's healthy. Somebody that's ill might be prepared to take the risk of side effects because they're so ill they want to get better, whereas somebody that's actually perfectly well needs to take into account that they may get some side effects. But the risk of disease, and clearly a disease like meningitis or pneumonia kills millions of children and older adults every year. And so I think that particularly the vaccines that I'm talking about, the benefits far outweigh the risks.
Okay, so a next question, a bit more COVID related. I know that's not the focus of today's session, but still revolving around vaccines. The question is, was herd immunity unachievable for COVID due to the scale of the disease? What are your thoughts on that?
So it's not about how many people it affects, it's about the ability to induce immunity that protects against transmission. And the great success of the pneumonia and meningitis vaccine, so the one against Hib, the first vaccine I showed you, the one against the pneumococcus, the one that I was talking about, and also the vaccine against meningitis that most people in the UK, or many people in the UK, have now received, is that those were able to generate so much antibody that they reduce person-to-person transmission of the bug and therefore generate herd immunity. The problem with the COVID vaccines is that they've not been able to reduce person-to-person transmission sufficiently to induce sufficient herd immunity. There may be some indirect protection or herd immunity, but really not big enough at this stage. So it's about the particular bug rather than about the volume of disease. And the diseases that I'm talking about kill far more people than COVID and affect far more people than COVID. These are much more impactful diseases. Obviously, concentrated within 2 or 3 years, COVID's had a very major effect. I don't underestimate it, but we're talking about diseases that cause many more millions of diseases and deaths in the population.
I think that's an interesting part, I guess, of the COVID pandemic. In some ways, it's almost bringing to light these other diseases that have existed for decades, hundreds of years, and actually have a much larger burden of disease across the globe. So I think hopefully this is like part of the conversation that's moving forward and taking more recognition of those, like you mentioned, pneumococcus and influenza.
And these diseases have, to some extent, been a bit of a casualty of people's concern about COVID vaccines because, and also, the lockdown measures that were put in place, because there were definitely decreases in the uptake of vaccines during the COVID period. And we have seen some increases in disease. I mean, initially, particularly in countries like the UK and some European countries where there was a lot of lockdown, actually that reduced person-to-person transmission and actually reduced the amount of disease, which was a good thing. But of course, as soon as people start to come back together again, as soon as people start to not wear masks, then these diseases become much more prevalent in unvaccinated populations. So, that there is, that risk in that sense. So there has been some harm to the vaccine programs as a result of COVID and people's concerns about COVID vaccines and vaccines in general.
That's really interesting. I mean, I wouldn't have anticipated that, but it makes sense in the way that you explained it. All right, so last couple of questions. The next one was, do you think there are any improvements when taking pre or probiotics or a more varied diet and thus a more varied and robust microbiome? And I'm guessing it's as a preventative method or protective method against some of the diseases that you mentioned.
So there's not strong direct evidence of taking probiotics or to improve what the bugs that are colonizing the upper respiratory tract or indeed the gut. There's no doubt, though, that if you disturb the bugs that are in the nose or in the gut with antibiotics, then that can predispose you to certain bugs that may cause disease. So disturbing things is not a good thing. But one of the things that we've been exploring is introducing bugs that are harmless but may interrupt the colonization of the upper respiratory tract. So the bugs that get into the nose that cause disease. So we've been exploring this in populations, or at least our colleagues in Southampton have been exploring this in populations in the UK, and then in collaboration with them, we've been exploring this in populations in Malawi. And it appears that we're able to get these bugs into the population. So the next question is do they interrupt these more harmful bugs from colonizing and being carried by susceptible individuals? So there's not strong evidence that taking yogurts or probiotics or any of those sort of things prevent disease. But I think the you're on the right lines, that if we can prevent the bugs that cause disease from gaining hold, then that will prevent disease.
I think that's pretty amazing. The use almost of weaponizing what's out there to help us and our betterment. I think that's pretty incredible. And you've touched upon lots of different kind of things that are in progress within the vaccine kind of ecosystem and technologies that are coming out and approaches that we're looking at. And I think someone's asked a really important question in the whole space of the development, which is, with the rise of artificial intelligence, for example, OpenAI and ChatGPT, do you see that artificial intelligence will be contributing or is already contributing to the production of new vaccines?
So I talked about looking at the genetics of bacteria, and there's no doubt that our ability to use supercomputers to analyze the genetics of the bugs that cause meningitis and pneumonia has revolutionized our understanding of how disease is caused, but also to be able to track bugs, because not every bug is exactly the same, although it might look exactly the same, but its genes are different and it behaves differently. And also then to potentially develop vaccines. And in fact, there's a term coined called reverse vaccinology, whereby by studying the genetic code of bacteria, you might then invent vaccines that then might cause disease. And that's certainly been used against meningitis, for example. So there's no doubt that these new technologies will accelerate our approach to developing vaccines, no doubt.
Yeah, I think it's like you said, just the sheer power that they have to process the volume of data, especially genetic data, like you touched on, is something that we can't do as physical humans, just one mind, one calculator. The computers are definitely a big tool that are going to hopefully see being a big part of the production process. So I'll ask you the final question before we close up for today's session. And it was around what we were touching on earlier, lower income countries. And we're asking, how are lower income countries overcoming financial barriers to providing vaccines at such large scales, or thereby how are developed countries aiding in that effort?
Okay, so that's a conversation that could take several hours, but I'll try to summarize it. So I think the answer is that there's a huge international effort, I have to say, championed by the Gates Foundation. So the Gates Foundation has been transformational in this space, in providing vaccines for the poorest populations, and that includes Malawi, and that's what the Gavi Alliance is about. The problem is the vaccines are incredibly expensive, and the real trap that's arisen is that as countries become richer, so as they transition from being the lowest income countries to being middle income countries, they can no longer afford the vaccines because they're getting less financial support. So, for example, if you take Kenya, which is transitioning from being a low income country to a middle income country, or South Africa, which is now a middle income country, they're going to have to pay for their own vaccines. And so things are becoming more problematic for countries, some countries in Africa, particularly countries in South America, which have some financial capability, but don't have the capability of the richer countries. The richer countries are all contributing to the vaccine effort. But as you saw from the COVID pandemic, when the chips are down, it can become every man. And I have to say every man, because it probably wouldn't be every woman if the women were in charge, but it's every man for himself. And suddenly the vaccines become available to the richer countries and not available to the poorer countries. And there was a huge inequity in the availability of COVID vaccines to the poorest countries, including in Malawi. So it's a complex issue. There is a real effort to try to protect the most vulnerable in the world, but I think there's a real trap for those graduating countries that are becoming richer and then being penalized and therefore not being able to afford the vaccines. And that's why, for example, people are looking at reducing the dose of vaccines and seeing whether they're still effective, because then that makes it cheaper, or the number of doses of vaccines.
Yeah, like you said, I'm personally very fascinated on this. We could talk about it for hours, but unfortunately, we only have 1 minute left. So I will wrap it up. And I will just say one more time, thank you so much for joining us today, your presentation, answering those questions for us. It was really, really interesting and very fascinating. So yes, like I've mentioned, we are out of time, but if I could ask the audience, please, to share your feedback with us today about the session. There'll be a survey coming out. If you could just fill that out for us, and that would be really greatly appreciated. Also, we will be having another fascinating lecture titled Tuberculosis, Technology and Global Health. This will be taking place on the 21st of March with our very own Hanif Esmail, Clinical Associate Professor in Infectious Diseases at UCL. The week of this timely lecture marks the lead up to the World TB Day on the 24th of March. We would love to see as many of you there as possible, and the details to that event will be passed out after this, prior to the registration. One last comment, we had a question regarding access to the lecture slides afterwards, and yes, I think Nef has already responded, but we will be recording the session today, including the slides, and these will be available on our website and the Faculty of Medical Sciences YouTube channel following the event today. Once again, thank you to the audience for joining us today and your wonderful questions and comments throughout. And once more, Rob, that was really great to have you here today, and thank you. Enjoy your evening, everybody, and take care.
Thanks, Ed. Thank you, everybody.